My name's Peter Searson. I'm a professor in material science and engineering in the Whiting School of Engineering at Johns Hopkins University. And I'm also a professor in biomedical engineering at the School of Medicine, and I'm a member of the Sidney Kimmel Comprehensive Cancer Center. In today's edition of Nature Nanotechnology, I talk about the role of in vitro cancer models in meeting the goals of the cancer moonshot. Because most people know the goal of the cancer moonshot is to significantly reduce the incidence of cancer over the next 25 or 30 years or so. In general, our knowledge of cancer and the way we advance detection and treatment and so on is based on studies in animal models and also with in vitro models and computational models. This is in part due to the fact that our ability to do experiments with humans is obviously fairly limited. In the past, in vitro models, although they've played a fairly substantial role in advancing our understanding of mechanisms and discovery of new therapies, drugs for example, have been relatively unsophisticated. However, over the last decade or so, there's been a tremendous increase in the complexity of these models, and we're getting much closer to being able to mimic the local microenvironment of a tumor in the human body to be able to better understand disease mechanisms and to develop new therapies. So advances in vitro models have been largely based in the fields of tissue engineering and stem cell technology and in microfluidics. And combined, these new advancing technologies have contributed to dramatically increasing the sophistication of in vitro models. We're now getting much closer to be able to mimic a growing tumor in tissue and even in the proximity of blood vessels, which are obviously crucial for the dissemination of tumor cells from solid tumors. These models are now poised to be able to dramatically improve our understanding of how these tumors grow and how they're disseminated throughout the body. At the same time, these models also provide us with much more high fidelity models to be able to determine the efficacy of new drugs and treatment strategies. In addition to that, microfluidic models and other related models are also dramatically improving our ability to develop high throughput detection and screening tools. With these advances comes the question of how can we harness these new technologies to have maximum impact and to contribute to meeting the goals of the cancer moonshot. The essence of what I'm talking about in this commentary is what we need to do to be able to achieve that goal. In my view, there are three main needs to be able to do that. And these are accuracy, qualification, and validation. So accuracy is establishing the ability of a model to recapitulate a particular facet of a tissue or a disease. Qualification is the demonstration that a particular model recapitulates a particular facet of disease in a particular context of use. Validation involves establishing that that model can be used reproducibly in different labs. Over the last decade or so, these advances I've been talking about have been occurring in individual labs, and all the models have slight differences and are developed in slightly different ways with no rules or guidelines in terms of producing the models or in meeting these requirements that I talked about for accuracy, qualification, and validation. So in order to make sure that we can harness the potential of these models, I think it's critical that we develop a framework and policies to be able to establish accuracy of a limited set of individual models for particular or specific context of use cases. So this may be a particular solid tumor type associated with a particular organ, or assessing the toxicity or efficacy of a particular drug for a particular cancer type. We need to establish qualification, that is, that the model is doing what it's supposed to do in this particular context of use and can be reliably counted upon to do this. And then finally, these limited set of models need to be validated. That is, we need to know that a single model will generate the same results in independent labs. I think it's crucial then that if we don't start to figure out these policies and procedures for these processes, we will not be able to harness the tremendous advancements that have been made over the last decade in meeting the goals of the cancer moonshot.